Going. We're going to get our PowerPoint up here. And today what we'll be doing in urinary system, last time we kind of got into just the big picture of things, right? What are the organs that make up the system? What are some of the general functions and things like that? We also started to understand that the kidneys are the big player here, right? That a lot of the other organs are just about moving things through or temporarily storing urine, etc. But urine is actually going to be produced in these kidneys. And as we were looking at their internal anatomy just at the end, we were seeing that they have a pretty complex vascularization in there, lots of different blood vessels. Proportionally, they get more blood than a number of other organs, especially of their size. And the pattern of the blood vessels, as we were seeing it, is they seem to start branching out and trying to reach this outer region, which is called the cortex. So something critical is going to be happening out there in the cortex. And so just like we see the branching of the bronchi to the bronchioles to the alveoli, and that's where all the action's happening, now we're seeing these blood vessels reaching out there as well. Now, the other thing that we mentioned about the kidneys is that is one set of tubes that we have in the kidney that are conducting blood through it. But in addition to that, we have a separate set of tubules in there that are going to be kind of running in parallel, kind of like the lymphatic with the cardiovascular, right? Where when we pull this fluid and some of these solutes out of the blood, they're going to route into the separate set of tubules and things like that. So we don't quite see any of those yet, but we will coming up. Okay, so just as a review as to what we're seeing there, right? We're seeing some larger structures as well. In fact, we can even bump back to something like this, right? So here's our kidneys. We have two paired kidneys. They have a renal artery going in. They have a renal vein coming out. So those are some of the largest blood vessels that are connecting to our abdominal aorta as well as to our inferior vena cava. So we saw those when we did our cardiovascular system. The kidneys itself, we saw that they have this little indented portion here. So that's a bit like we see in the lungs, right? So there's a hilus or hilum, an indented portion where major blood vessels enter in and out. But even on this really large level, we see big blood vessels, big tubes, and then there's a separate tube that we see right here. So the beginning of that is going to be what's called the renal pelvis. So just like the bony pelvis is going to be this area, kind of a larger area that has a collecting things, whereas our bony pelvis is collecting a lot of organs, urinary organs, including the bladder and intestines and things like that. Our renal pelvis, right? Renal means kidney, remember, is going to be an area where we're collecting the urine that's being created in the kidney. And we'll see that coming up a little bit more on the inside. But from that renal pelvis, which is going to be in here, we have the ureter, right? So the ureter is the other tube that we see connecting there in the renal hilum. So again, major tubes that are connecting and going into and out of that kidney there. So then we look on the inside of it. Now we're seeing, here's kind of that zoomed up version. So there is that ureter leaving out. And this expanded area here is the pelvis. So that is, on a macroscopic scale, our other kind of tubular system where we are conducting a fluid, in this case urine, that's been pulled out of our cardiovascular tubes instead. So this area that we see through here, this broad area, that's going to be our pelvis there. Now, in addition to that, we see the kind of the kidney tissue from here to here, and it subdivides into two different areas. I'm just going to switch over to like a green color that may be more obvious. So from here to here, we have the kind of what's called parenchyma or kidney tissue, and the deeper area is called the medulla, and the more superficial area is called the cortex. So we can see some of that. There's a term there in terms of cortex. And then there's a renal medulla like this that we see here. So those are regions of the kidney. Okay, just like we saw with the brain and the cerebrum, there was a cerebral cortex that's more superficial, etc. And on the outside, we have what's called a renal capsule. So there's a renal capsule or a fibrous capsule right on the outside as well. Some connective tissue wrapping to the outside of the kidney. So now we're starting to see kind of different areas of the kidney. You'll also notice that in this region that we call the renal medulla, we have these somewhat triangular structures here. And so they will historically call these lobes, but they're also called pyramids. So these triangular structures that we see right here are called pyramids, and those are structures that are found within the region that we call the medulla. And you'll notice that the point of those pyramids, like we see here and here and here, drain into this collecting area in the center that we were talking about as the pelvis. But those points are called renal papillae. So papillae, we had seen some of those structure or that term before. There were dermal papillae when we were talking about the skin and stuff like that. So the papillae is the point of the pyramid. And in fact, those pyramids are going to be places where we're starting to collect this urine. We're bringing it from the cortex where we said, oh, something's going on out there as our blood vessels are branching out there. After we form the urine out in the cortex, we're going to be kind of bringing it down deeper into the kidney through these pyramids, draining from the papilla, and then that's going to be collected by these branching structures, which each one of these is called a calyx. So you can see the term there, C-A-L-Y-X. So there's like these funnel-like structures. Each one's called a calyx that collects that urine, or if you want to think about it otherwise, almost like the coffee pot that collects it from the cone-like filter that's up above it, right? So these calyces, these little branches here, are collecting that newly formed urine and then bringing it to the pelvis, and then in the pelvis in here, we're going to drain it out through the ureter down to the bladder itself. So we've got some structure, some overall structure of our kidney there. And the other thing that we wanted to take a look at was, what was I looking at there? I'm trying to just going to get rid of some of those things there. Is I wanted to look at, erase, bink. Is I wanted you to look at the different blood vessels as well, right? So you can see that there's a number of blood vessels as we're starting going into the kidney, arteries going into the kidney. Renal artery is going to branch, and we get into these that are called segmental arteries. And you can see that these are going out to different areas of the kidney itself. Segmental arteries then become interlobar arteries. So just like we saw before in the cardiovascular system, when a blood vessel branches or it moves into a new area, it gets a new name, right? So we see it moving into an area between these lobes that are also called pyramids, right? Sometimes those are also called lobes. So those blood vessels going in between the lobes are called between lobar. Interlobar arteries. So we have interlobar arteries that are going to be sneaking through here like this. Then we go to an arcuate artery. So an arcuate artery, notice that we get to this line here between the cortex and the medulla. And it's like those blood vessels are afraid to enter into the cortex, right? So they kind of curve and go along the edge of it and form this arc. So those are our arcuate arteries that we see here. So an arcuate artery is going to be along here like this. So then that's the arcuate artery, and then that leads to all of these. So if we highlight these like this, you may think that they look kind of like the rays of the sun, right? That are radiating out into the cortex, which is that superficial region. So these are called cortical radiate arteries. So it's pretty straightforward, right, in terms of name, very descriptive. So those are finally getting the blood out to the cortex there. Sometimes those are called interlobular arteries because where they used to call these lobes, these little smaller regions out here between the arteries and veins were kind of like little lobules, right? So lobes, lobules, kind of like artery, arterial, and stuff like that. It's just another name for cortical radiate arteries. So cortical radiate arteries are going to get them right out to the cortex there, and that's where the magic's going to happen. That's where we're going to find these microscopic units that are called the nephrons that are actually going to be producing the urine. But we've got to get the blood out there to them, okay? So we're going to get the blood out there to these little filtering units, these little urine-producing units called the nephrons, and we're going to filter out a small fraction of the fluid that's in the blood, right? Some of that plasma. Now, I always think about it like, you know, if you have a pool or something like that, you have a filtration system associated with the pool as well, right? Because you want to maintain that large body of fluid in terms of its concentration and its cleanliness and things like that, right? So with that, what happens is you've got some sort of filtration system where it pulls out a small volume of the overall volume and then cleans it, right? Runs it through a filter, pulls things out, and then it returns it to the main body of fluid. So when we get to these little filtering units called nephrons out in the cortex, we're going to pull out a little bit of that fluid, and then the rest of the blood is just going to continue back via the veins. So when we went through these different arteries that I highlighted on the right, you'll notice that there are analogous veins, right? So there's a cortical radiate vein that's now routing this blood back from the cortex. There's an arcuate vein that's along the edge between the cortex and the medulla. There's an interlobar vein that goes between the different lobes of the pyramids as we're routing back here to be finally drained by the renal vein, okay? So this is part of the blood flow in the kidney. And as we move further, and as you're looking at your exam objectives, oh, we want to do that. Maybe this weekend, start looking at the urinary, right? Exam objectives. You want to be able to do this, what we might call tracing, right? So this might be a question that we could ask you on the lecture exam, like trace a molecule of water that we find in the plasma from the time it enters the kidney until it gets filtered out of the kidney, right? Until it passes into what we would call the urine. Or if it was blood that wasn't getting filtered and it was kind of following its way out of the kidney, you could then use those veins and things like that to kind of show how those different blood vessels are connected. So you can see going in, right, we have our abdominal aorta, renal artery, segmental artery, interlobar artery, arcuate, that curved one, cortical radiate artery. Now look at this little section in here. This little section that's at the bottom is showing us the microscopic structures. And to a certain extent, from what we know of the cardiovascular system, it kind of makes sense. Look, we have big arteries. They're getting smaller and more numerous. And then we got to arterioles. So you can see that there's an arteriole. We go arteries to arterioles. And this one is called an afferent arteriole. So that makes sense. A smaller artery, we call an arteriole. Afferent, you may recognize that from when we were doing nervous system, where there was sensory neurons coming in. They were also called afferent, right? Motor neurons coming out were called efferent. So we're seeing those terms coming again. Afferent arterioles are coming in. They are coming into this filtering part of the nephron, which is called the glomerulus. So the glomerulus is a very specialized set of capillaries that does the filtering that we think about as we were thinking about like doing coffee or something like that, where it's going to keep big things in the blood, like blood cells and platelets and large molecules and stuff like that, and it's going to let small things drop through, okay? So that's good. We go artery, arterial, and then glomerular capillaries, or just the small capillaries, bless you, can also just be called a glomerulus. All right, so that makes sense. So then we go, oh, look, arterial, capillaries. After the capillaries, we learn that it usually goes to small veins, venules, and then veins. Now here's where it gets a little bit wacky. What is that? We go from capillaries to an arteriole, okay? So the efferent arteriole is coming out of the glomerulus. Afferent arteriole is going into it. So this is a key place. This is where our filtration, one of our 3 different processes that we listed last time, is occurring, right? So we had filtration, and this is often called glomerular filtration. So that's where we pull these solutes and fluids out of the blood. Then there was, we said, reabsorption. That was our second key process in the kidney. And then there was secretion, putting things back into the blood, or into the filtrate from the blood if we needed to get extra amounts out. So in the glomerulus, we're going to do that filtration. But what we're seeing there is that it's not just a regular capillary bed. It's a specialized capillary bed that is about filtration. It's not about like where we usually have capillaries where it's oxygen and carbon dioxide exchange and it's waste products coming in and maybe nutrients going out and oxygen going out and maybe carbon dioxide coming in. It's not that type of capillary, okay? This is a specialized capillary bed that is exclusive to the kidney that is for forming urine instead. Okay, so we have the glomerular capillaries that go to the efferent arteriole, and now we see, and notice the color change if you're not colorblind, no offense, that's purple. We've gone from red to purple, And that's where we're suggesting, oh, okay, now we're getting oxygen exchange for carbon dioxide. We're getting maybe nutrients going into the kidney tissue to keep those cells alive, getting rid of their waste products, exchanging fluids, all the normal things that we saw about capillaries in the cardiovascular system. That's kind of what we're seeing with the peritubular capillaries. And peritubular, we said that we have a second set of tubules that are carrying the filtrate away that we've actually now made in the glomerulus. So peritubular, these are capillaries that are surrounding the tubules, okay? Because when we are talking about filtration, filtration was occurring in, I'm going to get a darker pen there, that was occurring in the glomerulus. So now we're linking together that structure with this function. That filtration occurs in the glomerulus, okay? So, but reabsorption and secretion occurs between the tubules and the capillaries. And we'll get more specific later on, but this is between the tubules and the capillaries, and in fact, what we're doing here is we are sending water and solutes and things like that from the tubules back into the capillaries. We are regaining it. We are reabsorbing it. For secretion, it's also between the tubules and the capillaries. But in this case, this is where we have extra things like hydrogen ions or something like that that we want to get rid of, or waste products, and we're going to push it using ATP and pumps from the capillaries into the tubules. Point being is that those other two processes that are so very important for making urine, in addition to filtration, occur between tubules and capillaries. So we have to have peritubular capillaries surrounding those tubules so that we have an exchange point. Just like in the lungs where we had alveoli surrounded by capillaries, so we had an exchange point, right? So that's what the peritubular capillaries are there for. Oxygen, carbon dioxide, all that normal type of exchange, but also the rest of this kind of renal function in reabsorbing and secreting solutes and water, etc. And we'll talk about that coming up. Okay, so from those capillaries, which are more like the regular capillaries, then we go to like venules and veins. And so you can see how it hooks into cortical radiate veins and arcuate and work its way out of the kidney there. Okay? So we have two sets of capillaries. The first one are called glomerular capillaries, or the glomerulus, and that's where filtration occurs. That's why that is there. These capillaries are more of your regular capillaries, but they're also responsible for doing this reabsorption and secretion, this movement of solutes and water between the tubules and the capillaries. Okay? All right. So for the most part, this starts to get us to a point where we can see why we're getting the blood kind of out to the cortex, like we were seeing. We're getting out to these glomeruli. These are only out in the cortex. And so that's why these blood vessels are working their way out to the cortex so that we can do that filtration, we can do that reabsorption and secretion. And then, once we're done with that, we can start routing the blood again out of the kidney itself and back into regular circulation. Okay, so these are just kind of examples of what you might do if you were doing tracing on like an essay question or whatever. You probably wouldn't have to do the whole thing, but maybe one side of it, like we said, starting from the point where we enter the kidney until it gets filtered out, or if it was something that was like reabsorbed or something like that, until it exits the kidney, like the other side, the venous side of it instead. Okay. So we got down from these macroscopic large blood vessels and larger tubes like the ureter and the calyces and stuff like that. We're getting down to this microscopic level, and this is where all the magic happens in producing urine, and it is created by this structure, the nephron. So the nephron is going to be your key player for making urine. This is a microscopic structure that consists of both blood vessels and tubules that actually does the work of creating urine. They're found mostly in the cortex of the kidney. There are little parts of them that extend out into the medulla as well, but they're mostly found in the cortex, and they're the ones that are doing the job of making the urine. So when we were at a bigger level, we said, oh, kidneys make the urine, bladder doesn't, ureter doesn't, stuff like that. Kidneys make the urine. Now we're knowing, oh, not all the kidney, just these little microscopic nephrons out in the cortex. And there's about 1 million of those per kidney, doing the job of making sure that the blood volume and composition is maintained. Okay, so then what is this nephron? We have this really important structural and functional unit of the kidney. It's kind of like when we were doing muscles and we're like, oh, sarcomere. That's the structural and functional unit of the muscles that's doing all the work of causing the muscle to shorten and lengthen, right? This is what's doing all the work of the urinary system in terms of producing urine. But what's it made out of? Just like in the sarcomere, we're like, oh, okay, well, there's Z lines and there's thick filaments and thin filaments. You had to understand what was going on in that functional unit. Same thing here. And so the components of this nephron is going to start with our glomerulus, right? Because that's where we're doing that filtration. So we have a glomerulus, which is that filter. And then we have surrounding the glomerulus, it's called a glomerular capsule. And the glomerular capsule, let me see if I can give you a shot right there. A photo, if you will. Sorry, a figure, not a photo. This here is the glomerulus, the red part, that's the ball of capillaries, and then this yellow part is the glomerular capsule. So if we're filtering things out of the blood, like a coffee pot, if you're filtering things, you better have something to catch that filtrate, right? If you're brewing coffee and there's no coffee pot down there, there's going to be a problem, right? So the coffee pot, if you will, or the part that's going to collect the filtrate when it comes out of the blood, is this surrounding structure called the glomerular capsule. So the glomerular capsule is around the glomerulus. Now, one thing I would highlight, when we were looking at the kidney overall, we said the kidney has a capsule as well, connective tissue capsule on the outside, but that's a renal capsule, kidney capsule, big one. This is glomerular capsule, just surrounding that ball of capillaries. So we see that we have our capillaries there, our glomerular capillaries, surrounded by our glomerular capsule. So the glomerular capsule is going to collect the filtrate that comes out, and then that filtrate starts its own little journey in those separate tubules. So you see this little windy part right here? That is called the proximal convoluted tubule. So if you look at that name, proximal, this means it's really right close to the glomerular capsule and the glomerulus, where the filtrate is starting from, where it's starting to be created. And then convoluted just means that it winds back and forth, right? Somebody's telling a story and they're like all over the place. You might say that that story is convoluted. It's not a straight line from beginning to end, right? So a proximal convoluted tubule is now taking that filtrate through. And then there's this interesting little loop here that goes down into the medulla. So here's our dividing line between the cortex and the medulla. And it's actually where those, what you're seeing there are the arcuate, artery, and vein. Right there, between the cortex and the medulla. So there is one part of the nephron that dips down into the medulla. It's called the loop of Henle, or it's also just called the nephron loop. That's maybe a little bit easier because it's like, I don't know, who is it, like Barbara Henle or something like that? I mean, great that they discovered it, but doesn't mean too much to us, right? So we do have this loop that goes down into the medulla and comes right back up again. And then notice that that tubule starts to get all windy and kinky again, right? So that is called the distal convoluted tubule because now it's further away from the glomerulus where we created that filtrate. So distal convoluted tubule. So those different parts that we were talking about there make up that nephron. Starts out with the glomerulus, where we're going to that ball of capillaries where we're going to pull the fluid out of the bloodstream. Then glomerular capsule, like the coffee pot that collects it. Then we start winding through these tubules, like the proximal convoluted tubule, the nephron loop, and the distal convoluted tubule, right? If you were not familiar with the kidneys and you didn't know the scale of what we were looking at, you might look at that and you're like, are we doing digestive system? Is that the small intestine again? Because I see this windy tube that must have some function, right? Why are we going to have it so long? Why are we going to have it, be kind of kinky like that, etc.? Well, just like in the small intestine, the length of that tube and the surface area that we have in that tube gives us that much area to now process this filtrate that we have filtered out of the blood, right? So the reabsorption and the secretion that occurs is going to be occurring in the proximal convoluted tubule and that nephron loop and the distal convoluted tubule. So just like we have to march our way through the small intestine to give it time and space to be able to absorb nutrients and digest foods and things like that, we've got to give our kidneys and our nephrons a bit of time to be able to pull back water that got filled out, to be pulled out nutrients and things like that that got filtered out, etc., right? Because that process that we talk about here, filtration, it's not about, ooh, I'm going to pull bad things out of the blood and I'm going to leave good things in. It's not that, right? It's all size selection. Just like the coffee pot at home, small things drop through the filter, big things stay in. There's a lot of very valuable small things in the blood, right, that's in the plasma that drops through that filter that we have to get back, right? A lot of that water, we said that these different nephrons, right, there's 1 million of them per kidney, 2 kidneys, they process about 200 liters of fluid, of plasma a day, which is 40 times more fluid than we have in our body. So the reabsorption, there's a lot of our plasma that is falling out through the filters, and we have to get it back. So it gives us the time to do this reabsorption and secretion. So we have those tubules that are going to give us the time to modify that filtrate, make sure that whatever we want to get rid of is in there. We've had time to push it in there or leave it in there. And things that ended up in the filtrate that we want to get back, glucose, all right, our important blood sugar or water, things like that, we're going to have a chance to pull it back and retrieve it before it gets lost as we pee it out in the toilet, right? We don't want to lose those very valuable things. Now, what this figure right now doesn't show you is that all those tubules are surrounded by capillaries, right? Paratubular capillaries. Because if we reabsorb something from the filtrate, where's it going to go? We want it to go back in the blood where it came from. So there are capillaries surrounding that. They just didn't put it in the figure because it's kind of hard. It makes it a little complex, but we'll see it coming up. Or if we're going to secrete, if we're going to push things from the capillaries into those tubules, they've got to be right next to one another, right? We got to have capillaries exactly with the cells and the tissues that we want to exchange with. Okay, and in fact, one other thing, take a look at this. Proximal convoluted tubule, these are showing the different types of epithelial cells that line these tubules. And look at the proximal convoluted tubule, that first section there. What do we see on those simple cuboidal cells? But microvilli. Just like we saw in the intestine, microvilli give us more surface area to be able to do the job they need to do. Yeah?
With all the things that go through that filter, would proteins be one of the things?
It depends on the size of the protein, right? So we do have, right, if you hearken back to when we were talking about blood, we're like, okay, we got to learn the different blood cells. We got to learn all the things that we find in the plasma, which is mostly water. But remember, we have 3 different types of plasma proteins. We got fibrinogen, we've got albumin, we've got our antibodies. All those things are critical that we need to maintain in the blood. Now, most of those are going to be too big to go through that filter. So that's good. So they just stay in there and they do their job and they don't get filtered out. But there are some other things that do get filtered out. Like we said, if we've digested proteins and we went down from protein to peptide and amino acids like we just got done learning, amino acids are super small. They'll go through there for sure. And so digested proteins will drop through there. Some we'll learn later on about the endocrine system. There are a lot of hormones that are made from proteins. And some of those hormones are really small, and they drop through there and end up down in the urine. So there are proteins that may be small enough to slip through the filter. But generally, by and large, our proteins are big enough that they won't fit through those little holes in the glomerulus.
So if they slipped through, then there was probably like some type of
damage. Kidney damage. We'll talk about urinalysis as we get to it in the lab. If you see big things showing through, like if you were making coffee and you pull the coffee pot and there's all kinds of grounds down there, you're like, what happened? That's not right. There shouldn't be any down there. Look at your filter, maybe a hole in the filter. Maybe it was overfilled and going around the filter.
Well, I asked because my husband has nephrotic syndrome.
Yeah.
So
Nephron.
His filters weren't working because he was losing albumin.
Oh.
So he swelled really badly.
Yeah.
So I just was trying to understand that more.
Right, exactly. So that's what we're doing here in our health science pathways is trying to understand how these things are supposed to work. So when something happens that throws them out of whack, you're like, wait a second, I know that there shouldn't be proteins in the urine. I know there shouldn't be red blood cells in the urine. So if I'm seeing that in my own or a family member or a patient, that should be a red flag. No pun intended, depends on the blood cell. But that should be some sort of a flag that something's wrong, that it's not working appropriately, right? Now what we'll find, and we're going to talk about this as we get into more of the physiology, is that if you remember when we were talking about our regular capillaries previously, we talked about how fluid, bulk flow, how fluid exchanges with those tissues, right? So we had hydrostatic or blood pressure, which pushes it out at the beginning of the capillary. And then we had, because of albumin, we have this osmotic pressure that sucks it back by the end of the capillary. So we have an exchange going on in regular capillaries. That blood pressure that causes that movement of things out of the capillaries is definitely going to be the same pressure that's going to generate filtration in these glomeruli. But it's going to be modified a bit, because what we want here to occur is not exchange. We only want outward movement. We only want filtration. Because we have these other processes that we have occurring down in the tubules. If we need to get stuff back later on, or secrete stuff later on, that's why we have the tubules. So in the glomerular capillaries, we only want filtration. So there's going to be something a little bit different. We're seeing something very unique about these glomerular capillaries. They're sandwiched between arterioles. That seems wacky. They're in a big ball as opposed to kind of spread out into a capillary bed that are more diffuse or something like that. That seems weird. So we're going to be taking a look at that a little bit closer coming up. But it's the first step in creating urine. It's not the only one, right? As we were talking about before you started to learn about the kidney, you would think, oh, yep, kidneys filter bad stuff out of the blood. That's what they do. But that's only one of three processes that we have to know about. Okay, so let's take a little bit closer look. Right now what we're doing is we're kind of looking at the different parts on a microscopic scale, and then we'll talk a little bit about their function and kind of migrate through, and then we'll go back and talk more about these three processes and how they work. So it's kind of like when we did respiratory system, remember? Right? We went through and we did anatomy, macroscopic down to microscopic, where all the magic happens. And then we said, okay, but what are the processes that have to occur for these organs, for these structures to do their job? We're going to do the same thing. Okay? Okay, so let's look at this glomerular capsule, which is kind of like the coffee pot. So I'm just bumping forward. This is the information that I'm going to be talking about on this slide that we see over here. Okay, so here is the afferent arteriole. Afferent, like we said, is coming in. That leads to the glomerular capillaries, which is in here. And then the efferent arteriole is going out. Now remember that even though filtration is happening here, we're not pulling out all of the fluid from the blood, right? Because if we did that, then what would be going into the efferent arteriole is just cells and proteins and dust and stuff like that, right? We have to have fluid that are still going through. So just like the pool, we don't empty the pool all at once and dump it into the filter and then put it back. We take a little bit at a time, regularly, in order to maintain the big volume itself. So we're going to filter some of that out, and when we filter it, it's going to drop into that glomerular capsule. There's our coffee pot like that. Now, the glomerular capsule itself actually has two layers to it. I think about it like the membranes, like the serous membranes that we've been learning about through time as well. So if you remember back to the heart, again, cardiovascular system, we've got the heart, and it has serous membranes associated with it. It's got one right on the surface of the heart. You guys remember what that was called? Visceral pericardium. Right? So we had a membrane that's right on the surface of that organ, and then we had the parietal layer of it that lined the pericardial sac, right? Lining the cavity. So if we kind of take that and we apply it here, the glomerular capsule also has kind of two layers to it. It's got a visceral layer that is right on those glomerular capillaries, and then it has a parietal layer that's out here that kind of is the coffee pot itself that's collecting that fluid that comes out. So the visceral layer of the glomerular capsule, so you see parietal layer of the glomerular capsule, visceral layer of the glomerular capsule, like that. The visceral layer of the glomerular capsule is made up of these cells that are called podocytes. So pod means basically, right, so when you talk about ped and stuff like that, you're talking about feet. And somebody thought that these kind of look like feet with toes or something like that. And the toes, and if it creeps you out, I don't know, maybe you'll remember it better, but the toes basically wrap around that capillary, and it helps to form the filtration membrane. It helps to form this permeable barrier where certain things, small things can get through, but bigger things are going to be retained. So that's what you're seeing there. That yellow layer are these podocytes making up the visceral layer of our glomerular capsule, and they wrap around it. So again, imagine like a foot with toes that are able to wrap around it like that. That's what we're seeing here. So these are little toes of each one of those foot cells of the podocytes, and they come to form that filtration membrane. So this little slit that we see there are filtration slits where our solutes and water are going to be able to leak through. Now also look at the underlying capillary there. Right? That's a capillary. That's a glomerular capillary. Look at it. It's a fenestrated capillary. So that's one of the types of capillaries we learned about before. We talked about continuous capillaries. We talked about fenestrated capillaries that have windows in them. And then we talked about sinusoids, right? Where we've seen these along the way. Like sinusoids, we saw them like in the liver, right? In between those. So especially leaky because we're moving red blood cells, etc. These are mid-grade leaky, because we want things to drop out of the glomerular capillaries. So we put holes in it so it makes it easier for things to drop through to create that filter. All right, so we're seeing a number of structures, both of the glomerulus and of that glomerular capsule, at least the visceral layer of it, that are making up what's called a filtration membrane, okay? So when we were doing lungs, we had a respiratory membrane, right? The wall of the alveolus, the wall of the capillary, and then a fused basement membrane. That is what we had to cut across for oxygen and carbon dioxide. Here, we have a filtration membrane, and the filtration membrane is also made up of part of the capillary and then part of some other cell that's on the other side of it, right? But in this case, it's always going to be an outward movement. We're always going to be having things leave the blood, not go back in, okay? Okay, so that's why that one's called filtration membrane. That's what we're looking at there. That's where our different solutes and water are going to be filtered out of the blood. All right. So questions, any questions so far in terms of what we're seeing, microscopic structures in the kidney, parts of the nephron?
You said solutes and water are filtered out?
Yep.
Out of the blood in this area?
Exactly. So our first process, that filtration, is the movement out of the bloodstream, and this is driven, we'll talk about it, driven by blood pressure, and that's going to push it out in that way. Okay, so that is our introduction to our glomerulus with its surrounding glomerular capsule. Then we have the rest of that tubular system, right? So the renal tubules are going to be our proximal convoluted tubule, starting with, and this is our first chance. After we've created filtrate, and as we said, there's a lot of valuable things in there, we want to hurry and get it back. Otherwise, we're going to lose it. And so that's why our proximal convoluted tubules are the ones that have those microvilli. They have a lot of mitochondria as well, because if we're doing secretion, reabsorption, we're pulling it across, right? Secretion is usually going against the gradient, and so this usually requires energy. So the proximal convoluted tubule is our first place to hurry get stuff back that might be lost into the urine. So this is the primary place where reabsorption and secretion occurs. It's kind of like, again, small intestine, primary place where we're doing absorption, but then what do we have afterwards? We have, it was the large intestine as well. So further down the way, we still have options of pulling other things in if we need it, okay? But proximal convoluted tubule is our first place where we're going to be doing reabsorption and secretion, so we have even more surface area there by having those microvilli in that section. Then we have our nephron loop and our distal convoluted tubule, other areas where we can do secretion and reabsorption. Now that nephron loop is a wacky thing. We see how it goes down into the medulla and then comes back up again. Seems like it makes no sense. Why do we go all the way down there if we're going to come back up? But it does have a key role to play in allowing us to produce different concentrations of urine. Because it would be one thing if we're just like, okay, filter stuff out, bring stuff back, like a static type of amount, so we can make sure, but we want to be able to adjust the kidney function based on what's going on in the rest of the body. So if we're dehydrated, if we're overhydrated, the kidneys need to adjust their activities accordingly. And so a lot of that ability relies on that weird-looking nephron loop. So we'll talk about that coming up. Okay, so this is just another view, again, of our nephron. Starting out here, proximal convoluted tubule, there's our loop, distal convoluted tubule. You'll notice that this whole thing, that's a nephron right there, that whole thing connects to this big tree-like structure, which is called a collecting duct. So a collecting duct is going to be collecting the urine that is produced by the nephron, by multiple nephrons, and bringing it down towards the calyces and down towards the pelvis. Okay? So a collecting duct is a little bit, again, kind of like our large intestine, larger diameter. Most of the work's already been done by the nephron, but by the time we get there, we might still be able to do some tweaks. A little bit, do we need to reabsorb a little bit more water? Do we need to reabsorb a little bit more electrolytes, things like that? We can do that in a last-ditch effort as we're going through our collecting duct. But once we get to the renal pelvis and the ureter and stuff like that, we're done, right? So whatever it is at that point, that's urine, and we're done processing it. Now it's just about storing it and getting rid of it. Okay? Okay. So this is, like we were talking about, in the collecting tubules, in those tree-like structures, we do have some ability to do some last-minute tweaks, including adjusting the pH of that fluid and helping to do some last-minute tweaks in terms of water and electrolytes. Okay? Very similar, like I said, to what goes on in the large intestine. Less surface area, bigger diameter, not as much going on, but we've got some options. Do we need to pull some things back more, or do we just let it go? We've got options at that point. Now, I won't get into, right, the difference between principal cells and intercalated cells. There are two different types of cells there, but function's important, that in the collecting tubule, we can do a little bit more water reabsorption, a little bit more salt balance, and a little bit of helping to maintain pH, right? So we're talking hydrogen ions there as well. Okay. So now we've kind of gone through a basic structure of our nephron. We talked about how multiple nephrons dump into those tree-like structures, which are collecting tubules, which have a chance, like the large intestine, to do some last-minute tweaks. The other thing that we want to address here a little bit is the fact that we actually have two different types of nephrons. We saw that kind of, it was like a surprise thing like we saw in the cardiovascular system where we're like, there's cardiac muscle cells. Wait a second, there's two different types of cardiac muscle cells, right? It's not going to be as detailed as that, but there are two different types of nephrons. And if we look at the terms there, we can even figure out what the two types are. So cortical nephrons are going to be those that are the most common and have most of the nephron itself in the cortex. So they do have a loop that goes down to the medulla, but it only goes down a little bit. So again, it's mostly in the cortex. Whereas about 15%, the remaining 15% of our nephrons are going to be juxtamedullary nephrons. So they have a nephron loop that deeps very deep down into the medulla before it comes up. And we said that the role of that loop is to allow us to make urine more concentrated if we need it. And so those that have longer loops are responsible for our most concentrated urine that we can produce. Okay? So if you're super dehydrated and you're only putting out a little bit of urine, it's going to be really dark because we have reabsorbed a lot of the water from it, and we're just adding a little water to flush out some of those metabolic waste products and things like that that we need to get rid of. So we're conserving water as much as possible. And those nephrons, with the especially long loops, have a big role to play in terms of us being able to produce concentrated urine like that. Okay? So here you can see an image of that. Remember, this is our border between cortex and medulla. So all this is cortex out here, and then medulla underneath here, right? And so a juxtamedullary nephron has a really long nephron loop, whereas a cortical nephron still has one, but it's just a little bit shorter. Okay? So they all produce urine. They all can produce concentrated urine, but those with the longest loops are really much more important when you need to produce really concentrated urine. The urine that we can produce is up to 4 times as concentrated as the fluid, as the plasma that we pulled out originally. So it's very good at conserving water if we need to. And if you think about it in terms of organisms that are really worried about conserving water, right? So things that live in the desert, they are definitely concerned about conserving water as well. If you've ever heard of like kangaroo rats and stuff like that, super cute. They have like really big long legs and they jump and stuff like that, but it's a little mouse-like, they call it a rat, right? But it's about the size of a mouse. They have many, many more of those types of juxtamedullary ones because they basically don't drink anything. What they do is they get their fluids from whatever they're eating, plant material and stuff like that. So whatever they're, if they're only taking in a little bit, they have to be very conservative with those fluids. And in fact, they have nephron loops that are so long, they extend way down even towards the pelvis, like deep down into the inside of the kidney, which means they can produce even more concentrated urine than we can. But it just highlights the role of that loop in allowing us to produce concentrated urine, okay? Okay. So this slide is just reviewing for us the fact that we have these 2 different types of capillaries associated with the nephron. The first one is the glomerulus, where we are doing filtration. And the second ones are those capillaries around the tubules where we are doing our reabsorption and secretion, our other 2 critical functions for the kidney and the nephron. Okay? So we just got to remember that we have 2 different types of capillary beds, and each capillary bed has a different role to play. Okay. Now, one of the other things, if we're talking about blood vessels and we're talking about blood pressure and things like that, we said that blood pressure is going to be just like in regular capillaries, it's going to be the primary driver of that movement out of the bloodstream. So filtration, like we were talking about. And the interesting thing about our nephron and our glomerulus specifically is that, like we said, we want only outward movement of fluid at the glomerulus. And one of the key ways that we do that is that we maintain the afferent arteriole, the one going in to the glomerulus, is always, if it's working properly, a larger diameter than the efferent arteriole. Okay? So the afferent arteriole is larger than the efferent arteriole. Let me pop back here really quickly to this shot. You may not have noticed it when we were going through this, but if you look at this, look at the size of this tube coming in compared to this tube going out, right? So the afferent arteriole is much bigger diameter than the efferent arteriole. What does that mean? That means that there's more blood being delivered to the glomerulus than it can drain at any given time. So that's going to create a backup, right? So think about maybe your shower tub or something like that at home. You've got an inflow pipe, like the afferent arteriole. You got an outflow pipe, like the efferent arteriole. What happens when your drainage pipe gets clogged? Hair, all kinds of nasty, weird, black stuff. I have 2 girls, right? I got 2 girls and 2 boys. I can tell you that drain, so bad. And then they won't clean it. They're like, no, I can't do that. This is disgusting. I'm like, yeah, it's disgusting and it's your hair. What the heck? But the point is that when that drain becomes clogged, if the diameter gets smaller, what happens? It creates a backup, right? Now in the bathtub, that sucks because now you're taking like an ankle bath and a shower at the same time. But in the glomerulus, that backup, that increased pressure, creates more filtration. It creates a pressure that causes the filtrate to come out of the glomerulus. And it's so large that we don't even have a chance for any of that fluid to sneak back in. In a regular capillary, it's like, oh, the beginning, it goes out, and the end comes back in. In this case, we have much more blood pressure in the glomerulus because we have this large inflow pipe, or afferent arteriole, compared to our drain. And so where is that extra fluid going to go? It's going to jump out. It's going to jump out into that glomerular capsule there. So it creates that filtration pressure so that we always have this net outward movement of fluids and solutes from the capillaries, the glomerular capillaries there. Okay? Okay. So we'll come back to that. That's a key aspect as to how those glomerular capillaries are specialized, that we have filtration occurring there. This is just showing some microscopic view of what we have been describing. We'll actually see this in the slide coming up. There is a ball of glomerular capillaries, right? That's what we're seeing right there. There, surrounding it, that white space, that is the glomerular capsule, right? So that's where the filtrate is going to be collected, right here. And there will be an afferent arteriole coming in and an efferent arteriole coming out, but through here you can actually see the different types of tubules as well as they're winding back and forth. So there are our proximal convoluted tubules, these ones that are a little bit more kind of closed in like this. These are proximal convoluted tubules. The things that are on the inside here that kind of close them off, these all have microvilli, right? So microvilli like this means that the lumen is going to be much smaller and it's going to have a fuzzy border to it as opposed to distal convoluted tubules. Here's an example of a distal convoluted tubule. A much wider open lumen and a nice clean edge to it because we don't have those microtubules. So in the microscope, we can actually tell the difference between proximal and distal convoluted tubules based on their structure like that. But all of those are where we're doing that further processing of the filtrate. That's where the filtrate was created, right? Filtration. This is where our secretion and reabsorption is going on. Still out there in the rest of the nephron like we were talking about. And this one's a scanning electron micrograph that again shows those balls of capillaries. And in fact, here you can see like an afferent arteriole coming in and a small efferent arteriole going out. And there's our ball of capillaries. So you can see that there's a lot of surface area in those capillary beds to allow that filtration to occur as well.
How did they get that photo? Was it one of those fancy microscopes where it like takes a picture? There's a term for it, I don't remember.
So this one, this is a scanning electron micrograph. So you can see things a lot smaller. So it uses electrons, right? Remember neutrons and protons, electrons? It uses electrons to image whatever we're trying to see. So rather than a light, like a light microscope, right? It's using light waves. Now we're using electrons. And a scanning electron microscope rains electrons down onto it, and then they kind of bounce off. A little bit like an X-ray, right? You get this reflectance. So you get the contours of it. You get this 3D nature of it because you're seeing the surface of it like that. And you can see things a lot better with a lot more detail, right? When we look at the light microscope, you can see things, but you're like, I don't know, that's just a ball of pink stuff, right? But now you can actually even trace, right, an individual capillary. You might be able to follow that through there. You just get a lot more detail.
Yeah, that detail's so interesting.
Yeah, it's crazy. So that's a scanning electron micrograph. There's also a transmission electron micrograph where the electrons would pass through instead, but you wouldn't quite get that 3D nature of it. Okay, so here we're still looking at, like we were talking about, that glomerulus. And this is just a close-up view of what we were discussing, the fact that our afferent arteriole has this much larger diameter here compared to our efferent arteriole, right? So a smaller diameter efferent arteriole is going to create this backup in the glomerulus there. And so that is what generates what's called glomerular pressure. Glomerular pressure is blood pressure, but it's going to be at a much higher level compared to our regular capillaries. And so rather than seeing it drop off and having fluids come back again, we're always going to have, like I said, that net outward movement creating the filtrate, which is exactly what we want to happen. And then when the filtrate exits the glomerular capsule, it starts its way through those tubules. And we can see those microvilli and the proximal convoluted tubules as we're going to do that reabsorption and secretion there. Okay? Now notice as well, there's a couple other interesting things here. There's some big tube right in the center of this glomerulus with its afferent and efferent arteriole. This right here is a section, the last section of the nephron loop. It's kind of this transition between when it becomes a loop and then it becomes the distal convoluted tubule. But notice it comes right back by the glomerulus. And so that's not an accident. I mean, we do see that they're all packed together, for sure, but it actually has a purpose. This part that we see right here of that nephron loop helps to make kind of a physiological checkpoint. All of these cells right here make up a structure that is like a little intrinsic monitoring system for these filters. So if you're thinking about, I don't know, this is like waste treatment, right? So you have lots of different waste treatment plants around and stuff like that. And what do they have? They have untreated, right, effluent and stuff like that, and right, water and extra things in there that are coming in, right? And they have to treat that so that when it comes out, you've taken out the chunks and various things that are in there. And then what do you have? You have something that is cleaner that you can use to irrigate plants or something. You're not going to drink it, not that clean, but it's processed it in that facility. And so based on if you have a lot of waste coming in, it's really concentrated, you might have to do some tweaks at the waste treatment plant to make sure that you're still going to get kind of cleaner water coming out. So this area right here, that part of the nephron loop that we see there and some of those cells of the afferent arteriole make up a very specific structure that's called the juxtaglomerular apparatus, but it's a checkpoint that we'll talk about in just a minute. Okay, so again, this is just highlighting some of those particular structures that are going to be involved in these processes, the peritubular capillary beds and the glomerulus. And then this is one that we haven't highlighted as of yet. The peritubular capillaries are going to be around the proximal convoluted tubule, the distal convoluted tubule, etc. But there's also these long, straight, right? So vasas, vessel, rectum means straight, like rectus abdominis and things like that. These are straight vessels that are wrapping those long nephron loops in the juxtamedullary nephrons. So they have the same job in terms of picking up solutes and things like that, but it's a specialized set of blood vessels that are extending around that nephron loop. So you can see them here. So those are vasa recta that we see along there. But this shows us a little bit more accurate detail about how all of those peritubular capillaries are surrounding our proximal and distal convoluted tubules, etc. Okay. So what we're seeing is that we've got a very specialized capillary bed called the glomerular capillaries that is going to produce filtration. As we've suggested, there is a difference in terms of how it is organized, where we have afferent and efferent arterioles on either side of it, right? And our afferent arterioles, much like a lot of regular arterioles, are protecting the glomeruli from big changes in pressure, right? Because remember when we were going through blood vessels before, we said the arteriole is like a big brother or big sister. It's got smooth muscle in it that it can clamp down on that blood pressure so that it doesn't blow out the kidneys that are very small and delicate, like a little brother or a little sister, right? So to a certain extent, our afferent arterioles are the same in that we're protecting glomerular capillaries. They have a job of making our filtrate, our urine, but we know that there's going to be blood pressure changes in the body overall, right? Maybe on exam day or lab practical day, your blood pressure is a little bit higher, right? We don't want to blow out our glomeruli in the kidneys and be peeing blood on a lab practical day, right? We have enough other things going on. So even when there's higher blood pressure overall, we need to make sure that the kidneys can still do their job, that they get enough blood pressure to do the filtering and stuff like that, but they're not getting too much because they have lots of capillaries that are really important and we don't want them being mistreated and allowing them to blow out, okay? So afferent arterioles are regulating the flow into our glomeruli, and then the efferent arterioles, again, we want to make sure that those are smaller than our afferent. So they help to maintain that high glomerular pressure, and they also then protect downstream from them. They protect the peritubular capillaries that are coming off of them. Okay. That was a small aside in terms of those, the purpose of those blood vessels there. But this is what I was just talking about in terms of that little physiological checkpoint. That combination of specialized cells from the ascending limb, the upcoming limb of the nephron loop, as well as from the afferent arteriole, they make something that is called the juxtaglomerular complex. It's also sometimes called the juxtaglomerular apparatus. So this is our physiological checkpoint where we can adjust the filtration that is going on in our glomerulus as necessary. So we can, in a few different ways, make sure that the glomeruli, which are doing filtration, are doing their job. Do we need to increase the filtration? Do we need to get rid of some excess fluid because we're drinking a lot or our blood pressure is too high? Or do we need to decrease the amount of filtration because we're dehydrated and we need to maintain blood pressure, etc.? So that physiological checkpoint, that juxtaglomerular, right? This is near the glomerulus, complicated thing, in essence, right? That is what is helping to control that. So it's like a little intrinsic control system. And as I was talking about, it's made up of parts from the ascending limb of the loop. Those specialized cells that are part of the loop are called the macula densa, okay? And those have chemoreceptors in them. It's a checkpoint. If you have a checkpoint at a waste treatment plant, maybe you have little windows there. You're looking at what's coming in and what's going out. You're like, oh, there's a lot of stuff coming in. I got to make some changes to how long we're going to process it and how much filtering we have to do, etc. Right? So we have chemoreceptors in those specialized cells called the macula densa that are checking the solute concentration. Okay, so that's important. If we're filtering the blood, we need to know, okay, blood pressure, how much blood do we have? We need to know the concentration of it, because the role of the kidneys is to adjust blood volume and blood concentration, right? That hematocrit, remember that hematocrit is supposed to be about 45% in terms of cellular elements to fluids? This is one of the ways that we maintain that, so the blood's not too thick, so it's not too diluted, etc. This is where we can work to maintain that. So chemoreceptors can, if you will, kind of check the concentration of the blood and see, oh, do we have too much fluid in there compared to our solutes or not, okay? Then we also have what are called granular cells or juxtaglomerular cells. These are the specialized cells that are on that afferent arteriole. And in these, they are mechanoreceptors. Oh, okay. So we can check the concentration of the blood, which is what we're supposed to be maintaining, and we can check the blood pressure. Because again, if we have too much blood or something like that, that exerts too much blood pressure. How do we get rid of that? Get rid of some of that fluid. Get rid of some of the fluid, blood volume goes down, blood pressure goes down. So what we have there are a couple of receptors that can then monitor the blood and see what we have to do, make adjustments as necessary, right? So just like any of those homeostatic loops, we start with a receptor, then we're going to have some sort of effector, right? We make a decision, we have some sort of effector to affect the change. In addition, we do have some secretory cells that will put out this substance, which is called renin, that we'll talk about later on, which is also kind of a hormonal or chemical control system for our urinary function. Now these ones are also our third set of specialized cells that make up our juxtaglomerular apparatus. They think that these ones may communicate then between our two groups of specialized cells, between the cells on the nephron loop and those that are on the afferent arteriole. So they're called extraglomerular mesangial cells. So everything that we're talking about is basically in here. And we're going to see it, sorry, extend a little bit that way. We're going to see it even on models coming up. So we have specialized macula densa cells that we find there. We have specialized juxtaglomerular cells or granular cells there. And then there's even some of those mesangial cells, the in-betweens there, okay? So that juxtaglomerular complex or juxtaglomerular apparatus is our little checkpoint. Because think about it, right? Afferent arteriole is raw blood, like raw sewage coming into the waste treatment plant. By the time we get to the distal convoluted tubule or the part of that nephron loop, then we've done a lot of treatment. We've done filtration already. We've done a lot of reabsorption and secretion already. So that's almost like it's almost ready to go. Let's check our product here and see if we need to make any adjustments, right? So that is the little checkpoint or that little window that allows us to do that. Okay. Filtration membrane, to a certain extent, we already addressed this, right? Like we talked about it, how it's the wall of the glomerular capillaries, the visceral layer of the capsule, as well as that fused basement membrane that are in between them. So we kind of already talked about that, so I'm going to jump over that. This part is a little small aside that I think is really interesting. So what we're seeing down the bottom here is that filtration membrane. So you're seeing there's the wall of the capillary at the top. The white layer in the middle is the fused basement membrane. And then you're seeing part of that visceral layer of the glomerular capsule down towards the bottom. There are, in fact, these little cells in here, part of our foot cells, remember? Our podocytes that are wrapping around it. Those toes, if you will, can actually change shape and change size. So that can affect the filtration membrane, right? Because here's our filtrate. It's going to fit through those slits and has to make it through the capillary wall in order to make it into the blood. Well, if we change the size of those, you can see how that adjusts how much is going to make it in between those parts of the filter, right? Whether they are elongated and we have big open sections, or whether they are kind of constrained there a little bit more. So those podocytes also have this contractile activity that can help to change the amount of filtration that's going on. So we're seeing different ways that we can tweak the amount of glomerular filtration that occurs. And in fact, this up here stands for glomerular filtration rate. Okay? So it's like this, glomerular filtration rate. So there's a lot of control of what goes on in the glomerulus because it's the first step in producing urine. So if we make tweaks here, then that affects everything downstream. Just like in the digestive system, whatever you're ingesting, that affects all the rest of the digestion, right? If you're eating more fat versus protein, how much you're eating, how little you're eating, how much fluid content there is and stuff like that, then all the rest of the system has to respond. So there's a lot of checkpoints and a lot of control that occurs right at the glomerulus because everything downstream then is going to be affected by that. Okay? So that's why we have our juxtaglomerular apparatus. That's why we have a few different other mechanisms that are going to control that glomerular filtration rate that we'll see coming up. Okay? Okay. So the last thing that I'm going to do is just review those three processes, just in general. And then we're going to kind of give you a little breather, right? Those that were up early doing lab practical, they can take a nap. Those that still have to do it can do a little bit of review, okay? So these are our key three processes: glomerular filtration, tubular reabsorption, and tubular secretion. So this very simple diagram here on the right is showing us a very simplified nephron. So up at the top here is our glomerulus. You know, the glomerulus is the capillaries. The glomerular capsule is the coffee pot around it. Technically, those two things together also have like a functional unit name that we'll talk about. But that's where filtration occurs, right? So filtration occurs up there. It's called the renal corpuscle when we combine those two things together, the capsule plus the glomerulus. But filtration only occurs at the glomerulus. So if we're talking about like our different processes for digestive system, we're like, oh, where does ingestion occur? Where does propulsion occur? Where does chemical digestion occur? Glomerular filtration only occurs in the glomerulus. Meanwhile, tubular reabsorption and tubular secretion occur between those other tubules, proximal convoluted tubule, distal convoluted tubule, nephron loop, stuff like that. They occur in the other tubular segments. And when we're talking about tubular reabsorption, by definition, that is going from the tubule back into the bloodstream. So from the tubule into those surrounding capillaries. Whereas tubular secretion is the opposite movement, from the capillaries, and we are using ATP often and pumping things into the filtrate into the tubule, because anything that stays in this tubule ultimately is going to be peed out as urine. Okay? Okay. So the reason that I'm highlighting those three is next time we're going to switch over from kind of all the anatomy and we're just going to focus on those processes. So let's talk about filtration, what it is, how we adjust it, and things like that. Reabsorption, and then secretion. Okay? Okay. All these are occurring in the nephron, but we have to understand the details. What are the forces behind them? What are different substances that are moved that way, etc. And that's what we'll do next time. Questions? All right, it's all about the nephron. Again, we're seeing, right, there are big organs, stuff like that in the system, but it's oftentimes the microscopic anatomy where the job is getting done. And in this case, we're talking about the nephron actually producing the urine that we discussed as a key part of the urinary system. Okay? All righty. Then morning lab people, you can take a nap, go get a snack, go over to Student Life and get a snack, free snack or something like that. And then later lab people, you probably have time for a snack, and then we'll see you next door.
How did you remember water?